Hello everyone. Thank you for giving me the chance to share our work on ISMB. The title of the paper is RiboDiffusion, Tertiary Structure-Based RNA Inverse Folding with Generative Diffusion Models. This work was a collaborative effort by a dedicated team of researchers including Huang, Ziqianlin, Dongchen He, Liang, and Yu Li. Huang is a PhD of Beihang University and a postdoc of CHK. Ziqianlin is an undergraduate of Nanjing University. Dongchen He and Liang are PhD students of CHK. Yu Li is an assistant professor of CHK CSE department, who is also the instructor of this project. You can access the preprint on bioRxiv and the code repository on GitHub. So let's begin. Nowadays, RNA design shows growing applications in synthetic biology and therapeutics, as RNA play a crucial role in various biological processes. Our work aims to explore the RNA inverse folding problem, which is an important part of RNA design field. RNA inverse folding problem is to find functional RNA sequences that satisfy given structural constraints. As shown in the figure below, RNA inverse folding tools can influence RNA sequences given secondary or tertiary structure constraints. In fact, researchers have already explored some methods to handle this problem. Most early computational methods for RNA inverse folding focus on folding into RNA secondary structures. However, without considering 3D structures of RNA, these methods cannot meet accurate functional structure constraints, since RNA secondary structures only partially determine their tertiary structures. The pioneering work Rosetta-based RNA sequences optimization applies a physically based approach to optimize RNA sequences and match the fixed backbones. But it is still constrained by the local design strategy and computational efficiency. Considering the shortcomings of these methods, an impressive idea is to apply deep learning to this problem. Thanks to recent advances in deep learning and the accumulation of biomolecular structural data, deep learning shows great potential in molecule design, in fields like protein structure prediction and protein structure design. Deep learning methods have achieved great performance. For example, protein structure prediction tool AlphaFold can regularly achieve accuracy competitive with experiment. Protein structure design tool RF-Diffusion can generate novel proteins that bind to target molecules. However, RNA inverse folding based on tertiary structure is still a challenging problem. On the one hand, RNA tertiary structure data still remains scarce compared to protein. On the other hand, this problem requires capturing the geometric features of flexible RNA conformations, handling the non-unique mappings between structures and sequences, and providing alternative options for different design preferences. As a result, we propose RiboDiffusion. First, we use RNA tertiary structures from PDB database to construct the benchmark dataset and augment it with predicted structures from the RNA structure prediction model. We split test sets based on RNA clustering using different sequence or structure similarity cutoffs. The key idea is that we formulate RNA inverse folding problem as learning the sequence distribution condition on fixed backbone structures using a generative diffusion model. Then, we also comprehensively evaluated the result of RiboDiffusion. We make a benchmarking related to sequence, secondary structure, and family similarity and analyze it across different RNA length categories and RNA types. We also explore cross-family performance, in silico folding validation, trade-off analysis, and data augmentation ablation study. So first, we would introduce how we use generative diffusion model in RiboDiffusion. As a first step of setting up the diffusion model, a forward diffusion process is constructed to perturb data with a sequence of noise. This converts the data distribution to a known prior distribution, for example, Gaussian distribution. Diffusion models can generate new samples starting from the prior distribution through learning to reverse the forward process. Such the reverse time denoising process can be described by a stochastic differential equation, SDE. In RiboDiffusion, we consider the RNA inverse folding problem as modeling the conditional distribution PS given X, where S is one-hot encoding for nucleotide types. X is backbone atom coordinates. In this reverse time SDE, this is the score function which represents the gradient field of the logarithmic marginal distribution. We establish a data prediction model to parameterize the score function, facilitating the reverse process. Following the typical diffusion models, we optimize the data prediction model with a simple weighted square error objective function. The architecture design of the data prediction model largely determines the diffusion learning quality of the diffusion model. We propose a cascaded model to predict the original nucleotide types, a structure module to capture geometric features, and a sequence module to capture intrasequential correlation. Our structure module is constructed based on the GVP-GNN architecture and adapted for RNA backbone structures. Our sequence module architecture is modified from the transformer block to inject potential conditional features. We construct a generative denoising process based on the parameterized reverse time SDE with the optimized data prediction model to generate RNA sequences that are likely to fold into the given backbone. We apply convenient ancestral sampling combined with the data prediction model and self-conditioning to generate sequences. During the generative denoising process, our model can balance the proportion of unconditional and conditional sequence distributions by adjusting the output of data prediction model. So here are the benchmarking results of our evaluation. We compare our model with 4 machine learning baselines with tertiary structure input, including geometric RNA design, pi-fold, struct-GNN, GVP-GNN. While geometric RNA design is a concurrent graph-based RNA inverse folding method, pi-fold, struct-GNN, and GVP-GNN are representative deep learning methods of protein inverse folding, which are modified here to be compatible with RNA. We also introduce RNA inverse folding methods with secondary structures and input for comparison, like RNA inverse, MCTS RNA, LRNA, and meta-LRNA. We use metrics including recovery rate, F1 score, and success rate, which can evaluate sequence, secondary structure, family similarity of generated sequences to native sequences. RiboDiffusion outperforms other methods regarding these metrics on different dataset splits. Then, we dive into a more comprehensive analysis of RiboDiffusion. To evaluate a cross-family performance, we repartition the dataset with the cross-family setting to verify the generalization of RiboDiffusion. We obtain the RNA family corresponding to the tertiary structure from RFAM database, then randomly select 4 families for testing and others for training. RiboDiffusion performs consistently in RNA families that do not appear in the training data. To verify whether RiboDiffusion generated sequences can fold into a given RNA 3D backbone, we use computational methods to predict RNA structures to obtain their tertiary structures. We take the TM score of C1 backbone atoms to measure the similarity between the predicted RNA structure of generative sequences and the given fixed backbones. Note that in silico folding validation contains two sources of errors. One is the structure prediction error of the folding method itself, and the other is the sequence quality generated by RiboDiffusion. As shown in the figure, RiboDiffusion generates new sequences that are different but still tend to fold into similar geometry. The performance for short-length RNAs is relatively poor, which is affected by the unsatisfied recovery rate of our model and the limitation of structure prediction tool itself. The benchmarking mentioned before consider the similarity between native sequences and the generated sequences. However, exploring novel RNA sequences that have the potential to collapse into this fixed backbone distinct from native sequences is still a realistic demand for RNA design. And there is a trade-off between the sequence diversity and the recovery rate of the generated sequences. RiboDiffusion can achieve this balance by controlling the conditional scaling weight. If conditional scaling weight is set to 1, the model only considers the distribution given the structure constraints. As we continue to lower the weight, we start to sample sequences from a mixture distribution of conditional and unconditional distribution, which can weaken the impact of structure constraints and improve the generation diversity. Augmenting training data is primarily driven by the scarcity and limited diversity of RNA sequences available in PDB. The incorporation of additional row-fold predictions improves the overall generated sequence quality. This augmentation also enhances the adjusting ability of RiboDiffusion for sequence diversity, where the sequence diversity of the model without the augmented data remains relatively low. So, in conclusion, RiboDiffusion is a novel generative diffusion model for RNA inverse folding based on tertiary structures. It has superior performance in benchmarking, robustness across families, well alignment for in silico validation, trade-off between recovery and diversity. In our future work, we aim to explore structure sequence joint design and RNA design in complex. That's all for my presentation, and thanks for listening.